Welcome to BIOL 102. Now, my dear students, this is the PowerPoint on the structure of plants, part 3. Today, I'm going to talk about the internal structure of root, stem, and leaf. So, let us now start with the first slide. And here in this slide, I am asking you I'm telling you that you already have an idea of different types of tissues, parenchyma, collenchyma, sclerenchyma. The only thing we are going to do now is we have to see how these tissues are arranged in root, stem, and leaf. And when I talk about these tissues in root, stem, and leaf, you have to remember the origin, location, structure, and function of these tissues. And then I'm asking you that you should be ready to answer question about the structure of root, stem, and leaf. Then it says when, and there are 3 questions marked. I think everybody got an idea when in quizzes, in midterm examination, in lecture examination, and during lecture, I also ask you questions. So you have to answer these questions when I ask you. The first of all, we have to study the internal structure of dicot root, monocot root, dicot stem, dicot leaf, and monocot leaf. Now you have to go to your lab guide also, because this is both lecture and lab combined. And in your lab guide, there are diagrams of internal structure of root, stem, and leaf. Find out these diagrams in the lab manual and ensure that you understand the internal structure of these organs. It will be a good idea to make diagrams and label the parts of root, stem, and leaf in your lab manual. In the lab examination and lecture examination, I will ask number of questions about these tissues and the internal structure of root, stem, and leaf. Try to do a good job in answering these questions and feel free to ask questions if you have any problems. Okay, now there are 2 terms, dicot plants and monocot plants. Dicot plants are those plants which have 2 cotyledons. Examples are chickpeas and pea and all the lentils that you leave. You are very much familiar with these dicot plants with 2 cotyledons in the seed. And monocots have only 1 cotyledon in the seed, and examples are grasses and lilies and palms and some of the other plants. So you should be familiar with these 2 terms. And here we are going to study the internal structure of dicot root, dicot stem, dicot leaf, and the monocot leaf. So these structures are studied in cross sections of root, stem, and leaf. So you should know what is a cross section. A cross section is one in which you have this root and you cut a section at right angle to the long axis. This is cross section or transverse section, and all these sections that you see are the cross sections. Okay, another 2 terms, primary plant body and secondary plant body. There are 2 types of meristems. Primary meristem, which are located at the tip of root and at the tip of shoot, and you are familiar with this stem tip and root tip. They are the primary meristem. Primary meristems have primary growth and they form primary plant body. All plants have primary plant body. On the other hand, there are secondary meristems which are present on lateral side, and they are responsible for secondary growth and they form secondary plant body. Wood and bark are examples of secondary plant body which develop from secondary meristems. Secondary meristems are not present in all plants. They are present in gymnosperms and woody dicots. Later on, you will study this secondary growth and secondary plant body. Right now, you are studying the primary plant body. Okay, so let us start with the internal structure of dicot root, and I will explain it with the help of this diagram of the internal structure of dicot root. The outside layer of the dicot root is epidermis, and epidermis of root has large number of root hairs, and one of these slide shows these root hairs. You can see that they are extension of epidermal cell, and there is a large number of root hairs on the root, and they increase the surface area, and their purpose is to increase to absorb water. They absorb water, so they play a very important role in the absorption of water. In this slide you cannot see the root hairs because of processing, but root hairs are present. Root hairs are present. So root hairs are very important. The root hairs are very important. Okay. Next to this epidermis is a big zone which is called cortex. Cortex is a big zone, and this cortex is made up of storage parenchyma cell. Cortex is made up of storage parenchyma cell, and you are familiar with storage parenchyma. They store the food material. Next to this cortex is this layer which is called endodermis. This layer is called endodermis. And endodermis is a layer which controls the entry and exit of substances from the cortex to the center, and from the center back to the cortex. And the center of the root is called stele, S-T-E-L-E. The stele in the case of dicot root is has star-shaped xylem in the center. So you have this star-shaped xylem with 4 arms, these red cells that you see with this lumen. This is xylem. This is star-shaped xylem. And in between the arms of the xylem, you have phloem. So this is xylem, and this is phloem, this is phloem, this is phloem, and this is phloem. So star-shaped xylem with phloem in between the arms of the xylem. And next, one of the important character of this endodermis is the presence of Casparian strips. You will see these Casparian strips in this slide that I will show you right now. These are the Casparian strips. The brown thing that you see are the Casparian strips. They control the entry and exit of substances from the cortex to the center, and from the center back to the cortex. So Casparian strips are present in this endodermis. Again, I will repeat endodermis, and next to the epidermis, there is a layer of parenchyma cells which become meristematic, and this is called pericycle. Pericycle gave origin to branch root or lateral root. Branch root or lateral root is formed from the pericycle. In the center, you have star-shaped xylem, and in between the arms of the xylem, you have phloem. So this is the structure of dicot root. I hope you understand this, and the bullet points that I have here also explain the internal structure of this dicot root. Here you have internal structure of monocot root. In the case of monocot root, again there is epidermis and there are hairs. Now remember that the hairs that you have in the case of root, they do not have cuticle, because they have to absorb water. There is no problem of loss of water, so they do not have a cuticle or a very thin cuticle. So here epidermis with root hairs. Again you have cortex, and cortex is made up of storage parenchyma. Next to this cortex is endodermis. As I have told you, endodermis has Casparian strips. Casparian strips are present in the endodermis. And inside the endodermis is the stele, and stele in this case is made up of xylem, which are these empty spaces here, and phloem, xylem and phloem, xylem and phloem, xylem and phloem. They alternate with one another. This white thing that you're seeing are the lumens of xylem, and this greenish thing that you're seeing is the phloem. Xylem and phloem alternate with one another. And the center of monocot root is occupied by pith. There is a pith which is made up of parenchyma cell. So this is how the structure of monocot root is. There is a pericycle under the endodermis. So epidermis, cortex, endodermis, and in the center you have xylem and phloem bundles which alternate with one another, and the center is occupied by pith. So this is the structure of monocot root. This one shows the origin of branch root. I told you that pericycle becomes meristematic, and this lateral root comes out from the pericycle. This is what it is. If you have carrots at home, you can cut a section of carrot and you can see this branch root coming out from the pericycle. This is a good diagram which shows the comparison of dicot root and monocot root. On the left hand side is dicot root and on the right hand side is monocot root. So you can see in dicot root there is a star-shaped xylem with phloem in between the arms of the xylem, and on the right hand side is monocot root which has pith in the center. So most important difference as far as you are concerned between dicot root and monocot root is the presence of xylem in the center in dicot root and the presence of pith in the case of monocot root. Okay, from root we can go on to the stem, and here again we have dicot stem and monocot stem. Example of dicot stem is sunflower, and example of monocot stem is corn. So let us first look at cross section of dicot stem. Here I have a diagram. Here you can again see a epidermis with hairs, and next to the epidermis is a narrow cortex. In this case, cortex is made up of two tissues. Just beneath the epidermis is collenchyma, which has uneven thickening of pectic material, and next to this collenchyma are layers of this parenchyma, and this cortex store food material. Now next to this are these structures which are called vascular bundle. Each vascular bundle is made up of xylem and phloem, and the arrangement of this xylem and phloem is such that phloem is toward the outside, toward the epidermis side, and xylem is toward the inside. And in between xylem and phloem you have a patch of meristematic tissue which is called vascular cambium. On the outside of the vascular cambium there is this patch of sclerenchyma tissue. This is called bundle cap. It is present on the outside of each vascular tissue. Now the important characteristics of this dicot stem is the presence of a large number of vascular bundles which are arranged in the form of a ring. You can see these vascular bundles which are arranged in the form of a ring. This you have to remember for dicot stem. And the center of dicot stem is occupied by pith. The center of dicot stem is occupied by pith. So this is the structure of dicot stem. There is another slide of the internal structure of dicot stem. You can see the epidermis, the cortex, the vascular bundle with the sclerenchyma on the outside, xylem toward the center, phloem toward the outside, and this is how these vascular bundles are arranged. Now we can go to monocot stem. In the case of monocot stem, example is corn. You can see that the outside of monocot stem again is epidermis, and whatever is present inside the epidermis is called ground parenchyma. It is called ground parenchyma. And in this ground parenchyma, there is a large number of vascular bundles which are scattered. So this is very important, arrangement of vascular bundles. Vascular bundles are scattered in the case of monocot stem. And here you can again see this monocot stem with the scattered vascular bundles. Here these are the vascular bundles which are like rods, and this is cross section of the monocot stem, and this is one vascular bundle. So it has V-shaped xylem. V-shaped xylem with phloem which is present in between the arm of the xylem. This is the arrangement of vascular bundles, vascular tissue in the vascular bundle, and the whole vascular bundle is surrounded on the outside by sclerenchyma. So this is the structure of monocot stem. Here again you have this monocot stem, and then as far as comparison is concerned, this slide shows very well the comparison of dicot stem and monocot stem. Remember, in the case of dicot stem, the vascular bundles are arranged in the form of a ring, while in the case of monocot stem, these vascular bundles are scattered. So this is an important feature you have to remember between dicot stem and monocot stem. In dicot stem, there is a vascular cambium. In monocot stem, there is no vascular cambium. Now we can go to the internal structure of leaf, and here we have a three-dimensional picture of the internal structure of leaf. In the case of leaf, it is a bilateral organ. It has upper surface and lower surface. All of you have seen leaf. So there is upper side of the leaf and there is lower side of the leaf. And this leaf is covered by this pink-shaped material made up of cutin. This is called cuticle. Cuticle will prevent the loss of water. In the lower epidermis, you have these pores which are called stomata, and each stomata has two guard cells. They control the opening and closing of the stomata. The gases exchange takes place through these stomata. So epidermis, upper side, lower side covered with cuticle, and in the lower epidermis there are stomata. The tissue that is present between the upper and lower epidermis, this is called mesophyll tissue. Mesophyll tissue is present between the upper and the lower epidermis. The tissue that is present just beneath the upper epidermis, this is called palisade tissue or palisade mesophyll. The whole tissue is called mesophyll. Mesophyll is of two types. Beneath the upper epidermis, this one which is barrel shaped, and this is called palisade. And the one which are irregular, this is called spongy mesophyll. So palisade mesophyll and spongy mesophyll, both of them are parenchyma. They have large number of chlorophyll, and all of you know that this type of parenchyma with large number of chlorophyll, this is called chlorenchyma, and photosynthesis takes place in this chlorenchyma. Okay, in this mesophyll there are a number of veins. If you have not seen structure of a leaf, look at a leaf and you will find number of veins. This is how these veins look like in cross section. And these veins have phloem, which is located toward the upper side, and the xylem, which is located toward the lower side. So phloem and xylem are present in the case of this vein. There is xylem and toward the upper side and phloem toward the lower side. So this is how they are arranged: xylem toward the upper side and phloem toward the lower side. So you can see this three-dimensional picture of the structure of leaf. This is dicot leaf. This is dicot leaf. Remember, in dicot leaf, the mesophyll is differentiated into palisade and spongy layer. But in the case of mesophyll leaf, again you have upper epidermis, lower epidermis with stomata. But in this case, mesophyll is not differentiated into palisade and spongy parenchyma. All mesophyll cells look similar to one another. So this is important difference between dicot leaf and monocot leaf. That is palisade and spongy in dicot, but in the case of monocot, the mesophyll cells, all of them are similar. Second, the veins are surrounded by prominent sheath cells. You can see these cells on the outside. These are called bundle sheath cells. Very prominent bundle sheath cells are present in the case of monocot leaf, but they are absent in the case of dicot leaf. So this is structure of monocot leaf. This one also shows the structure of monocot leaf, and this is structure of one stomata, and here these are the two guard cells with chloroplast, and this is the pore that is present in this stomata. So these stomata are very important because exchange of gases takes place through these stomata. So when I will talk about photosynthesis, I will talk about this structure of leaf and structure of stomata. Again, here he tells you about the comparison of dicot leaf and monocot leaf, and you can see that in dicot it is differentiated into palisade and spongy layer, but in the case of monocot it is not differentiated. All cells are similar, and double sheath cells are monocot in monocot are prominent, and they are not that prominent in the case of dicot leaf. So with this, I come to the end of this recording. I hope you have got a good idea about the internal structure of root, stem, and leaf. You've got a good idea about the internal structure of root, stem, and leaf. And this is the end of this recording. This is the end of this recording. Okay, let me see. This is the end of this recording.